speaker is Heather Smith from SCID Angels for Life Foundation.
Good afternoon. I'd like to disclose that SCID Angels is a paid consultant for Mustang Bio. My name is Heather Smith, and I'm president and founder of SCID Angels for Life Foundation, which I started 15 years ago. More importantly, I am the mother of 2 children born with severe combined immune deficiency, or SCID. SCID is the most severe form of primary immune deficiency, and in lay terms, these babies are born without a functioning immune system, leaving them vulnerable to infections as simple as a common cold. This is what happened to my first child, born before the era of SCID newborn screening. At the age of 6 months, he came down with what we thought was his first cold, but it developed into something much more serious. A rare form of pneumonia, which is primarily seen in patients with SCID and should have been a red flag, but unfortunately, the doctors didn't recognize this in time, and 4 weeks later, he passed away. In 1994, when I became pregnant for the second time with another SCID baby, we researched our options and learned the standard of care treatment for ex-SCID at the time was a bone marrow transplant. We also discovered that being treated at a hospital with expertise in SCID would require us to travel out of state. We weren't crazy about that idea, but we were willing to do anything to better the health of our unborn child. During that visit with the medical experts, we learned about an experimental treatment therapy that was successful in the animal model, but had not yet been done in humans. After reviewing the research papers and much discussion with our family, we opted for the experimental treatment, and in 1995, my son, Taylor, was the first in the world to successfully undergo a bone marrow transplant in utero while I was still pregnant. Taylor led a healthy and productive life, but during the spring semester of his junior year in college, we learned his immune system was waning, and it was only a matter of time before he'd have to seek additional treatment. Again, we did our research and weighed the risks and benefits associated with each option, but when your options are limited, like they are with SCID, you must make a decision based on the potential benefits foremost. That's exactly what Taylor did when he decided to enroll in the NIH lenti-vector gene therapy trial. 7 months following graduation from college, Taylor was inpatient at NIH receiving his gene-corrected cells shown here in this slide. As someone who has personally helped pioneer an experimental therapy and then gone on to assist her adult child in the decision-making process surrounding gene therapy, I've been through it and have a pretty good understanding what the issues are when it comes to the methods and approaches for capturing post-approval safety and efficacy data on cell and gene therapy products. 5 minutes isn't enough time to go through all the issues I've seen firsthand and feel need to be addressed, but I will take a moment to discuss one that I feel is the most important: the manner in which cell therapy participants are notified of an adverse event that has affected at least one participant under the protocol and triggered the suspension of a gene therapy trial is alarming. During a previous FDA public listening meeting held last November, I briefly spoke of this problem and asked for something to be written within the protocol guidelines, stating that if a safety rule were triggered, immediate notification to enrolled study participants would be made surrounding the circumstances before updating the clinicaltrials.gov website. This was because of an actual incident that happened to enrolled participants within the adult ex-SCID NIH lenti-vector gene therapy trial. In the summer of 2022, the SCID Angels private Facebook group received a post from a SCID parent saying it's been seen on the clinicaltrials.gov website that the trial has been suspended, and the reason in parentheses states clones representing 10% or more of the subject's patient's myeloid lineage have been detected or evidence of malignancy found. You become alarmed. It is my understanding that safety rules were written in the protocol prior to starting the study, and if one of those safety rules were triggered by a certain percentage of study participants, enrollment is stopped and the trial is suspended, which is what happened in the trial my son is in. Fortunately, there's no evidence in any of the study participants that there's an increased risk of cancer or any abnormalities in blood and immune cell formation or function at this time. Ironically, the gene therapy trial LVX-SCID-ND for newly diagnosed infants sponsored by St. Jude and using the same vector as the NIH trial, recently paused enrollment and was put on a voluntary hold at the beginning of April of this year for the same reason as the NIH trial that my son is enrolled in. This time around, the families were all notified by the PI before it was posted on the clinicaltrials.gov website as being suspended. I don't know if this is a result of my previous testimony or not, but I do know the participants, for the most part, were pleased by the written letter that was received and the informational fact sheets that were included. However, there are still improvements that can be made when something like this happens. The participants in the St. Jude trial are not being made aware of who the patients are that are showing clonal expansion, like we did in the NIH trial. This is extremely unnerving for most of the families, as they feel they have a right to know if their child is one of the 3 who have recently been uncovered. Finding the viral vector within the HMGA2 gene was first reported over 10 years ago, and to date, no patients with circulating cells containing an insertion into HMGA2 have developed any type of blood cancer. But my point is, transparency is crucial, and in this circumstance,
You're running out of time. Can you wrap up? And
and in all circumstances to come, unless you can promise that the blood cancer can't happen after the current 15-year-long follow-up study period has been completed, and unless follow-up studies turn into lifelong, we must be vigilant, those patients, but especially those who are currently showing clonal expansion. Please consider the way study participants are made aware of these types of suspensions. Thank you for your time.
Thank you, Heather. We understand that 5 minutes might not be sufficient time to share your perspective. We encourage all participants to submit their detailed comments to the docket.
Okay.